Hello everyone. Today we're going to do a case study. These are the Adhikaris. They are a fun little family where the youngest member is baby Shiv and this is his older sister Susie. As baby Shiv and Susie have been spending a lot of time together, Susie starts to notice that baby Shiv does something weird with his eyes where he keeps on moving them around. Initially, Susie thinks, well, that's just how babies are. But as time passes by, she has a feeling that something is indeed wrong. So she decides to take him to a doctor. Now, at the doctor's office, the doctor performs some tests and notices these additional symptoms. Baby Shiv has an increased sensitivity to light, a condition known as photophobia. Like Sus noticed, his eyes do move around a lot, a condition known as nystagmus. And finally, the outermost covering of his eye is abnormally thin and almost cone-shaped. You can see that if you compare it to the normal eye here, which has a very smooth circular surface, a condition known as keratoconus. These symptoms are very concerning, so the doctor decides to perform an electroretinography test, which is basically a test that detects the function of the retina. He then diagnoses baby Shiv's condition as Leber congenital amaurosis, also known as LCA. Leber congenital amaurosis is a genetically inherited family of disease that causes degradation of the retina in the eye. The onset of this disease is pretty early in life, as seen in baby Shiv's case, where vision progressively gets worse throughout the growing years, and it can lead to complete blindness. However, treating Leber congenital amaurosis is not such an easy task because this disease is not just a single disease, it is a family of disease, like a phenotype that could have been caused by varying mutations in at least 14 different genes in the body. If you look at this table here, it lists all the possible genes that could get a mutation leading to this disease. It also shows the mutation frequency in them and also lists their function in the wild type cell. Luckily, the doctor has gone ahead and found out that baby Shiv has a mutation in this gene here. It's called RPE65 gene, which is crucial for vitamin A visual cycle in the eye. Now, even within the RPE65 gene, there can be at least 21 different DNA sequence changes that can create a mutant RPE65. If you look at this table, we can see that there's a bunch of point mutations, and there's also some insertions and deletions causing frameshift mutation. In case of baby Shiv, he has this mutation here in the 56th position where a thymine has been replaced by a cytosine. Now, as a result, ATG is changed to ACG. ATG is our initiation codon that codes for MET, while ACG codes for threonine. Now, research suggests that this mutation leads to either no RPE65 protein being made as the initiation codon is substituted, or there are cases where a faulty RPE65 protein is made from a cryptic translation initiation site. This brings us to our next question. What exactly does RPE65 do? This chart here is a simplified version of our visual cycle that mentions actually a lot of things that we discussed in class. Rhodopsin is a photosensitive pigment in the retina that stays bound to 11-cis retinol in its inactive form. When light hits the retina, this rhodopsin gets activated to a new compound and releases that 11-cis retinol to form its isomer all-trans retinol. And if you noticed, this 11-cis retinol is an isoprenoid. Now, this activation event is the event that converts that light signal coming into retina into neural signal which can be sent to the brain. So you can imagine this is a very important event. Now let's focus on the byproduct, which was the all-trans retinol. That compound is acted on by a dehydrogenase and reduced to form all-trans retinol, which is again acted on by another enzyme to form this compound. It is an ester. Now this is where things get really important. The RPE65 protein that we have been discussing until now, it comes into play here. This protein is an isomerohydrolase, which means it isomerizes and hydrolyzes this ester simultaneously to form this 11-cis retinol. This 11-cis retinol, just like before, gets acted upon by a dehydrogenase, which oxidizes it into our initial 11-cis retinol. This is really important because this initial compound 11-cis retinol can now go and act on this activated rhodopsin and say, hey buddy, time's up. Let's go back to our inactive form so that we can receive new light signals. And that's how this entire visual cycle gets reset. Now let's focus on our mutation. What if we had a faulty RPE65 protein? It wouldn't work, which means this cycle would never be reset. We'd go from here and just make our way until here and produce this ester. But then we would have no mechanism to do this conversion here, which means every activated rhodopsin just stays active. It never is brought back to its inactive form. And no inactive rhodopsin means there's nothing to receive new light signals, which means no sense of vision. And over time, as there's more and more ester accumulation and less 11-cis retinol reproduced, this situation is what causes slow degradation of the retina. In our case, that's what we are calling Leber congenital amaurosis. Now, luckily for baby Shiv, the first ever treatment for this RPE65 mutation was approved by the FDA in 2017. Its name is Luxturna. It's a gene therapy that works by delivering a functional copy of RPE65 gene to the retina. So now there's like a functional gene, it can produce a functional RPE65 protein, which can go on to compensate for the effects of the faulty RPE65 protein that we initially had. Since it's a relatively new therapy in the US, more and more doctors are still adopting it. A quick search on Luxturna's website shows Susie that there is in fact one doctor in Richmond itself who is administering this new gene therapy. So the future for baby Shiv looks promising. He has a very viable option. However, not everyone is this lucky. Leber congenital amaurosis still continues to affect 2 to 3 children per 100,000 live births and still is the leading cause of inherited blindness in children around the world. So we still have so much work left to do in combating this disease. That's all I had for today. If you have any questions about this video at all, you can email me at susmita.adhikari@richmond.edu. Thank you for your time and thank you for watching this video.